Hi everybody, welcome back. We are at the first session of a new module for the honors program here. We taught this for the first time in 2018. A postdoc named Caroline Beltran put together a whole bunch of great slides on the subject of clinical biomarkers, and students seem to like it, so we're back for 2019, and I'm really grateful that you all signed up for it this year. So. I will be doing, I think, 5 of the 7 segments, and a postdoc from Stellenbosch University, Byron Reeve, will be supporting another couple segments. So we'll have a total of 7 sessions of class, as I recall. At the end of it, we have to have some sort of assessment, right? We can't just say everybody got perfect scores. So we'll do probably a multiple choice test that takes you through all of the lectures. All of the lectures will be represented in that final assessment. So I hope that you will be able to keep up to date with the class. As you see, I've got the slides here. The PDF of this is also available online. The YouTube video will have a direct link to that directory at the top line of its description. So hopefully that will make it very easy to keep up to date with what's going on in class. Grand. So welcome to biomarker research. I should explain that when I was a PhD student, I said to myself, well, that biomarker research might be very well funded, but I'm never going to get involved in that stuff. That's for crazy people. But in fact, biomarker research in clinical science is one of the most important areas where biotechnology can translate to direct benefits of human health. So eventually I was pulled in and spent 10 years at Vanderbilt University in particular working on biomarkers in colon cancer. So I will probably be making allusions to a lot of cancer work along the way. But that is not to say that biomarkers only have relevance in the cancer treatment. There are plenty of other ways that it plays into our lives. So we'll encounter several of those along the way. So we're going to start today with defining just what biomarkers are all about. This is a pretty neat encapsulation of the whole field, and I hope that this lecture gives you the structure to fit all the other topics into that are going to go into it. Eventually we're going to be talking about some subtle work in statistics and machine learning even to explain why those fields have had so much importance in the space of biomarkers. But for now we're just trying to set up a framework that all of these topics can slot into. We'll be trying to talk about what characteristics we expect in a good biomarker. What can you call a success as a clinical biomarker? We'll talk about lots of different types that exist in this space. Look at the differences between an anatomic biomarker or a molecular biomarker. Definitely talk about why single molecules or single biomarkers versus panels of biomarkers are preferred for making clinical decisions. Talk about the biomarker discovery pipeline, and look at lots of possible pitfalls that exist in this field. So this is a very wide-ranging individual lecture, but I hope that it will help set the context into which all the other pieces can fit. So what is a biomarker? Here we see that NIH and WHO have both given us some definitions. These are not the only definitions available from those bodies, so lots of people kind of differ on these points. But we start by saying that a biomarker is a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. Well, that kind of seems like it's making this term fill a whole lot of different roles, doesn't it? But we want to be able to measure something about a person generally, and from that we would like to be able to make some sort of clinical decision. So these are the things that we seek. Objective measurement means that we're not doing a do you feel better or worse today opinion kind of question. We're trying to get measurements that are about objective reality. So these are not really opinion feedback. You know, when we go to the doctor, they often say, where are you on the pain scale from 1 to 10? 10 is like organ failure, and 1 is like I have an itch I need to scratch, you know, there's a lot of room between there. But that's a pretty subjective way to go about evaluating how you're doing. So objective measurements are much easier for us to use as an input to clinical decision making. Okay, now the WHO suggested that any substance, structure, or process that can be measured in the body or its products and influence or predict the incidence or outcome or disease. Right, so we have a lot of, again, a lot of different ways that we would like to be able to use this information, but we note that we may not be measuring the body directly, but rather by products. So you might be working with biofluids that have been taken through venipuncture, or you might be working even with urine. There are lots of useful bits of information that we can get from the body and its products. So at base, we want to be able to make a measurement of a biological component where the analysis is able to tell us about disease state. All right, now if we've really got a great biomarker on our hands, we have one that's specific to disease. So I have a lot of friends who work in immunological biomarkers. They ask about cytokines. But one of the things that we measure in looking for tuberculosis, for example, is something like interferon gamma. But interferon gamma may rise in level not just due to tuberculosis infection, but in response to other infections, right? So that's an example where seeing an elevation in this might cause us to look at someone more closely, but you're not going to say it's tuberculosis just because this one molecule jumps. There's more information that's required for us to make that kind of determination. High sensitivity. Sensitivity is a really big deal. Now we talked about specific to disease. I'm going to go ahead and use the biostatistical term, specificity, right? That this marker rises in response particularly to this stimulus, right? That's specificity. Sensitivity is quite different though. We want to be able to detect at as early a stage as possible that someone has this disease. You know, right now our solid criteria for saying someone has tuberculosis is a pretty slow test. We have them cough up some stuff and see if we can culture tuberculosis bacterium, can culture it from their sputum. And that takes a while to do. Wouldn't it be nice if we could detect tuberculosis at a much earlier stage, long before they're hacking up viable bacteria? Okay, so high sensitivity gives us the best lead-up time, the earliest possible detection for disease. Safe and easy to measure. Well, I know some people do biomarker discovery in stuff like CSF. Does anyone know what CSF is? Cerebrospinal fluid. Right? So if you had a biomarker that was perfectly sensitive and perfectly specific for a disease, but it required you to get cerebrospinal fluid from someone, would you get a lot of volunteers? Not so much. On the other hand, most people have adjusted to the idea that if their doctor comes after them for a little sample of blood, they'll do it, right? Most people, if asked by their doctor to produce a urine sample, will do it, right? So these are examples of relatively non-invasive means, very safe means for us to do clinical testing. So something that is safe, a safe test, one that's easy to produce, is very good. If you require a biopsy from someone's brain in order to do your test, does this person have a disease or not? Not very safe, and certainly not very easy to get. Okay. You need something that's rapid for fast diagnosis. So if you're going to have a 2-month turnaround time from the moment you acquire a sample from someone and the time you can make a clinical decision about them, that's not very good. We really like overnight turnaround times, or, you know, it turned blue, you're pregnant, that sort of detections, right? Which is another thing to point out here. Not all biomarkers are about disease. You know, if somebody is hoping to grow their family and they want to know if they're pregnant or not, that's also a biomarker. We're not detecting disease, we're detecting pregnancy in that case. Okay, we need it to be cheap. All right, so I work with mass spectrometry. Does anyone know what a mass spectrometer costs? Maybe 10 million rand. Okay, so that is an example of an expensive detector. What about a PET-CT? Do you know what a PET-CT is? Big imaging device? An MRI? Yeah, expensive devices, right? So those are not ideal for cheap measurements, right? What we really like are things like ELISA, little lateral flow assays that allow us to detect by antibody whether a particular protein is present. That's dirt cheap by comparison to having to run a mass spec on someone's samples. Okay, consistent. A long time ago, I was part of a study at a group called the Association for Biomolecular Resource Facilities that asked how reproducible are the PSA measurements that we get if we send the same sample to multiple sites. You'd be surprised how much variability we can have in these bits of sample processing that just based on site-to-site differences. So consistent measurements where the same result comes back for the same sample time and time again is quite valuable. And eventually, of course, if you detect a difference in a particular molecule, it needs to have some clinical bearing on whether somebody is called sick or not sick, or pregnant or not pregnant. Okay, so the ideal biomarker needs to be able to cover different stages of disease. They need to be accurately and reproducible. They need to be accurate and reproducible. They need to be quantifiable. They need to be cheap. They need to be quick. They need to be consistent. They need to have clear effects on treatment and intervention. And ideally, they're significantly increased in the disease condition. If you get back a positive on your test, it's really quite important that actually, that the clinical situation you're trying to call is actually present. So I want to point out that there's essentially no clinical biomarker that meets all of these criteria. There's some sort of wart on pretty much any clinical test in existence. And we're always working on improving the biomarkers that exist for things. A very quick story. Do you know how we went about pregnancy testing early in the 20th century? It's a South African story. Has anyone heard of Xenopus laevis? It's a frog that lives here in South Africa. And they're now found all over Earth. And the reason is that Xenopus laevis was used for pregnancy testing for a very long time. Basically, you'd get a little bit of urine from a woman who wondered whether she's pregnant or not, inject it into the frogs, and if they ovulated, you said, ah, the lady's pregnant. Right, so that is an example of a clinical biomarker. Now, is it perfect? No, because I gotta say, lady frogs are not that keen on having urine injected, right? So, and those frogs are then not really useful for a few months for further pregnancy testing. Yes, some frogs went through multiple rounds of this. So, this is an example of how clinical biomarkers can progress to become better and better over time. Now, apparently women can pee on a stick, and if it turns a color, they're pregnant, right? So that's obviously a much more straightforward kind of testing. No frogs required. Okay, great. So, I think this diagram is one of two that I really like about this talk. So I want to make sure if you have it in your notes, you can just put a little circle around types of biomarkers. This is a complex story, so I'm going to try to tell it straightforwardly. Many different biomarkers, many different purposes for biomarkers may exist for an individual disease. For one disease, there are lots of different kinds of biomarkers that may apply. So predisposition biomarkers. What are the characteristics of a nominally healthy person that may predispose them to getting a particular disease? So it may be a genetic susceptibility biomarker. Someone close to me has celiac disease, for example. There's a genotype associated with this disease. That doesn't mean you have the disease, but it means you're much more likely to develop celiac disease than somebody who doesn't have the genotype. So a predisposition biomarker is not to say, are you now sick? It's to say, are you likely to be sick? Frequently, we find that predisposition biomarkers are quite associated with pedigree, that your family history in a disease is associated with one particular genetic change that makes it more likely for you to get this disease. So if you have a history of men in your family dropping dead of a heart attack at age 50, they may have what's called a widowmaker mutation in a particular gene, which makes it much more likely that heart attack happens at that age for men, right? It may be that you have a particular allele for the BRCA1 gene. Has anyone heard of this? BRCA1? BRCA1 is associated with breast cancer. If you have two of these cancerous types of alleles for BRCA1, it's very, very likely that you will develop this disease in the future, right? So these are examples of predisposition biomarkers. I would just say one of the easiest to detect predisposition biomarkers that we have for tuberculosis is smoking. If you're a smoker, you are far more likely, upon exposure to TB, to develop it. So that's an example of a non-genetic predisposition biomarker. Okay, so let's now move to what we think of as kind of the early detection case. How do you detect early on somebody who has subclinical disease, somebody who has already begun a disease process? So these are sometimes called trait biomarkers. So we want to determine or confirm the presence of a disease or disorder through early detection. A lot of people say to cancer researchers, why haven't you done your job? Why haven't we cured cancer? Right? We have cured cancer, lots of cancers. But the most challenging problem for us is to be able to detect at an early enough stage these cancers so that we can do something about them. You know, if somebody comes to their doctor for the first time with a 2-centimeter mass in their, inside their skull, that's problematic for us, right? It's very difficult to have any sort of magic cure that can address a problem like this at a very advanced stage of disease. So, the earlier you can detect a cancer, the better your shot of actually doing something about it. Lots of people, when they become sick in their 50s or 60s, are like, I don't want to go see a doctor, I'm old, of course I'm going to have a disease. Well, the reality is, if people are able to go to a doctor at a time when you can do something about the disease, rather than when it's spread all over their body and laced through their bones and so on, you have a much better shot of addressing the disease at that early stage. So early detection is frequently about this diagnostic biomarker condition. Okay, now, prognostic biomarkers. What does anyone know what the word prognosis means? Prognosis. Very much so, yeah. To prognosticate about something is to make a prediction about how it will go. So you could say, well, there's an allegation in the news about somebody taking money when they shouldn't have. You could prognosticate about that and say, is this person going to go to prison? Well, I know these factors about the situation. Is this person going to go to prison? They're not going to go to prison, right? Oh, they're definitely going to prison. They're fried, right? So this is a prognostication. In the same way, in disease, we like to be able to say, if we did nothing, would this person get worse or better? Lots of people self-cure, right? You know, I'm going to tell you a really scary factoid. Everyone in this room has had abnormal cell growth that could have caused cancer.
Thank you.
Now, we'll all sleep better, right? The reality, however, is that our bodies are really good at taking care of that nonsense. If a cell gets out of line, the other cells send out signals and the immune system rallies and says, oh, this cell's got to go, right? And that cell is dead. Our bodies develop problems all the time. And our immune systems and other systems are able to confront that problem and solve it. We have really powerful bodies that are able to cure themselves of a lot of things. So it is possible that although a condition exists, the body is well on its way to dealing with it. So we would like to know what the likelihood of a disease recurrence would be, or a progression might be. One of the situations I'm going to talk about a lot is Grandpa and the doctor. So Grandpa has been having some difficulty peeing lately. I'm going to talk about this because it's a clinical importance, right? So Grandpa's had some difficulty peeing. Grandma finally convinced him he has to go to the doctor, and the doctor says, my goodness, your prostate is large. So it could be that means Grandpa has prostate cancer, and it could be that it's an aggressive prostate cancer. But it is not necessarily the case that this is so. Lots of men with prostate cancer would never die of prostate cancer. Prostate cancer can be indolent, right? It can just sort of lie about and say, well, I'm going to have normal cell growth, but I'm kind of content where I am. Or it can be one of these aggressive monsters that goes racing throughout the body. Prognostic biomarkers would help us to discern which of these two outcomes, which of these two disease courses is most likely for a given disease. All right, Now, predictive biomarkers are useful for us in understanding which treatments are likely to be most valuable for someone. So let us say someone has TB. Does anyone know how we go about treating somebody with TB?
You usually take drugs for 6 months.
Drugs for 6 months, yes. Does anyone know what kinds of drugs?
Antibiotics?
Antibiotics, yes. Specifically, we frequently see isoniazid and rifampicin. INH and RIF, sometimes you'll see those names used. Okay, so that is one course of treatment. But will rifampicin and isoniazid be viable for solving everybody's TB?
No.
Definitely not. In South Africa, we've got some crazy strains of TB out there that are wildly dangerous. Some of them are completely resistant to treatment by rifampicin and isoniazid both. If so, we better know that from a predictive biomarker as early as possible, because treating somebody who's already resistant to isoniazid and or rifampicin with that drug cocktail is a really bad idea. Okay, so being able to predict whether a particular treatment regimen is viable or not is an example of a predictive biomarker. Finally, treatment response biomarkers. If you are treating someone for a period of 6 months for a disease, you would like to know sooner rather than later whether the disease is reducing in scope or growing in scope in someone. So, having the ability to detect whether the treatment is in fact working is a very important kind of biomarker to do. We'll be talking about some of those in just a minute. So, I hope that this helps to demonstrate that for any one disease there are lots of different stages at which you might want to learn something about clinical outcome. And many different biomarkers can litter this path from somebody who's just maybe apparently completely healthy to someone who is undergoing active treatment for a disease. Okay, so here we are using just the case of breast cancer as an example across all of these. So we can ask among people who have the BRCA1 and BRCA2 mutant alleles for these breast cancer genes, who has a predisposition for illness. We might use something like mammography as an example of how to diagnose at an early stage whether someone has breast cancer. Right? I mean, if you're completely unaware whether a person has or doesn't have any lumps in a breast, is a very fundamental piece of information for discerning who does or doesn't have breast cancer. The number of people who have breast cancer and no detectable lump whatsoever is pretty small. So being able to detect that through mammograms, through biopsies as well, or through a protein-based blood test, gives us these are all ways that different biomarkers that give us the ability to diagnose this disease at an early stage. For prognosis, we frequently make use of information on tumor size. It's a very simple kind of metric, but basically a pinpoint tumor and a centimeter or 2 centimeter tumor are going to get a completely different kind of response out of a cancer doc. The assumption is that if you've got a big tumor, you're in a much bigger world of hurt than a tiny pinprick. Lymph node metastases. Metastasis is a term that shows up a lot in clinical biomarkers. Does everyone know what metastasis is in cancer? It's not just about growth.
But spreading.
Spreading, right. Okay, so we often talk about primary and secondary tumors in cancer. So a primary tumor is where the cells first started going awry. But frequently we see that a primary tumor will send out cells through the circulatory system and they lodge in a new place and they create a new tumor there. That's a secondary tumor. So this process of spreading from a primary tumor to create secondary tumors is called metastasis. And frequently when we talk about cancer, this is associated with like a stage 4 diagnosis, although the terminology I think is a little bit inconsistent across cancers. So metastasis is a very bad idea. Lymph nodes are part of the lymphatic system. It's kind of like the body's cleanup filtering system. So if you find that metastases have shown up in lymph nodes, it's an indication that you have circulation of tumor cells. It's bad news. Okay, predictive biomarkers. A lot of people make the mistake of thinking that breast cancer, for example, that's the slide, that breast cancer is one disease. Because, you know, it starts in a breast and it's a cancer, therefore it's a breast cancer. The reality, however, is that different breast cancers can behave very, very differently depending on which receptors they're producing. Some of our best treatments for breast cancer depend on particular receptors being active and wild-type form in these tumor cells. If those receptors are not the standard type, and you try to use a targeted therapy designed for that particular receptor, you're going to get absolutely nowhere. It makes just as much sense as treating a virus with antibiotics, right? You're sending a message that the cells aren't getting. So we have good predictive markers in the world of breast cancer cells to determine which treatments are viable. Okay, treatment response. Often we see that biological imaging is used for handling treatment response to determine whether this course of treatment is reducing the tumor size or not. So that's this final example. Okay, so in the slide before this we were talking about them in text. Here we're looking at them in the explicit example of breast cancer. I hope that helps to, the example helps to demonstrate the concept a little more clearly. Any questions so far? Okay. I always feel kind of bummed when I think about cancer treatment too. I spent 10 years there, and I wish that I had a more cheerful feeling about how biomarkers are going as a field in the space of cancer. We get better all the time. So, what about a non-molecular biomarker? I often talk about things like a particular gene or a particular drug susceptibility test. These are kind of molecular markers. But frequently, we're using physiological and anatomical measurements. So if you're using something like PET-CT or MRI, you're probably trying to characterize non-invasively, but typically quite expensively, whether a particular tumor is growing or not. So some of these are, some of the measurements of this sort are very useful as a research tool, but much less useful as a widespread clinical tool, right? How many of the clinics in South Africa currently have a PET-CT instrument? Yeah, sure, the cities will have them, but if you're out in living in between cities, you may have to travel quite a long distance in order to get access to something like this, or have a medical scheme that will pay for it. That is another rather important detail. All right. On the other hand, there's a lot of investment now in the space of molecular biomarkers. So Mary Claire King and friends did a lot of work with breast cancer, the BRCA1 gene and BRCA2 genes. This is an example of a genomic biomarker. People may be born with the genetic variants, the alleles associated with getting breast cancer. There are little girls walking around right now who haven't even gone through puberty, but who have a much elevated chance of eventually developing breast cancer. And many of them will be completely blind to that until someday they discover a lump. You know, this is the reality. All of us have within our genomes many different alleles that could plausibly lead to disease. Or, you know, we may be walking around with the alleles that we could contribute to our offspring that could lead to disease. For example, cystic fibrosis. You know, if I were really planning on having children, I would like to know, if I had a family history of cystic fibrosis, am I myself carrying a copy of that gene that I could contribute to a child? You know, if that kid has 2 copies of the cystic fibrosis allele for that gene, that kid's going to have cystic fibrosis, and that's a difficult life. Yeah, so these predisposition biomarkers like that have these really complex family histories to them that are worth thinking about. Transcriptomic biomarkers are typically biomarkers that are built around gene expression right? So when we talk about BRCA1 and 2, we're not talking about is there a lot of it or a little of it being produced in a cell. We're asking what is the sequence of the thing, right? And if that sequence is the disease-associated allele, that's problematic. A transcriptomic biomarker, on the other hand, talks about gene regulation. If a lot of this transcript is produced or a little of that transcript is produced, it may have an effect on whether you are developing a disease. It may be that your body's gene regulation, gene expression, is changing as a function of disease state. So it could be that long before your doctor can detect that you have TB, your genes are already mounting a response to it through gene expression. Okay, proteomic biomarkers are a really big deal. Now, I'm a proteomics guy, so when I hear proteomics, I'm always thinking of a mass spec. As I've mentioned, a mass spec is a great research instrument, but it is not a great clinical instrument, typically. There are some sites that have turned it into a clinical instrument. They're very amazing. But typically, when we go hunting for a particular protein associated with disease, we're not doing so by mass spec. We're doing so through antibodies. So something like PSA, the prostate-specific antigen, is a good example of a proteomic biomarker that we can measure as an indicator of early disease. Of course, a lot of times we're interested in metabolic disease. I think many people know that South Africa is rapidly becoming home to a bit of a widespread worry about metabolic syndrome, which is to say that as people are eating more and more, as they're eating more Western food, we see that more and more people are getting that, like Americans. I'm just saying, I've gained a lot of weight in just a year. People who spend more time sitting in front of a computer and who are eating a lot can occasionally develop weird metabolic conditions. It may be that we have excessive blood and excessive sugar in our blood. And that sugar can modify proteins and do all kinds of badness that can lead to something like metabolic syndrome or diabetes. So these biomarkers typically are built around things like A1C measures, which are not measures of whether a protein is present or not, but maybe about how much various sugar structures on them are. Okay, sometimes we're interested not in individual molecules and not in the whole body, but about particular cells. So my family, for example, has a propensity to have very low platelet counts. Have people heard of platelets? They're very important for clotting, right? So in my family, the males at least, have a increased propensity to have too few platelets. So for something like that, we would want to measure cytometrically, to measure cells in blood. So these are a variety of different ways that we might measure for molecular or cellular biomarkers. All right. I think I've already mentioned prostate-specific antigen, the PSA test. It's our most commonly used method for screening somebody for prostate cancer. So that's an example of a single protein test. We have just this one protein we test for, and its quantity is used to determine whether somebody has an enlarged prostate and likely prostate cancer. However, there are lots of conditions under which PSA will increase in level. You may have an inflammation of your prostate. If you're a man, I mean, women don't really worry too much about prostate cancer. But an enlarged prostate will increase the amount of PSA in one's blood, even if somebody doesn't have cancer of the prostate. So you can see that there's a big weakness here in the simplicity of a one-marker test. As a result, we see that there are great problems in these single tests for detecting a variability of disease. It may be that a 10% increase in one person is hugely diagnostic, whereas a 30% increase in another is just normal variation. Dealing with heterogeneous populations is problematic. If you're dependent on one marker, you're quite likely to have lower accuracy, lower sensitivity, lower specificity. And you're going to be less able to detect all of the cases out there. So single biomarkers are of limited specificity and are problematic for this. Okay. Generally speaking, we see that people are moving to using panels of biomarkers. So instead of building everything about the readout from one individual molecule, we have a family of them that we measure as a cohort within an individual to say, is this ensemble together giving us indication that this person is positive for this disease, for example. A lot of this is fueled by the fact that we're in the omics era. As Al said, the omics error, that would be a little unfair to our field. We're in a biotechnology department here, so you're aware that we have lots of sequencing capability. We can measure the genetic variability in a person very broadly. We can measure the expression of individual genes through RNA-seq. We can measure all kinds of things. Proteomics certainly has given us the ability to measure thousands of proteins across cohorts of samples. That's great, and we now have a lot of computing power to make sense of that. But being able to combine lots of different proteins together or lots of different genes together to put together a single signature for a disease is actually quite difficult. We need to be able to have multiplex measurement, meaning we need to measure lots of biomarkers as an ensemble to make our decision about whether this person is positive or negative. We need to be able to we need to have some degree of independence in those assays that if these two genes always move up and down together, they don't have a lot of information separately from each other because you can predict the behavior of the other given the behavior of one. But having more more lines of evidence to combine is actually a whole lot better for us in achieving better sensitivity to detect someone as positive when they are, and specificity, being able to correctly say someone is negative when they're not. Being able to build all this information together is quite challenging. So ultimately, getting to a place where we are able to make many measurements about an individual en route to diagnosing them positively or negatively, or saying they are responding to treatment or they are not responding to treatment is critical. Personalized medicine will happen through biotechnology coupled with a lot of wisdom about which of these measures fluctuate quite a lot in their own regard without respect to the disease, and understanding how a person's individual genetics play into the phenotype that we can measure at a molecular level. So I'm going to, for now, when I talk about panel biomarkers, I would like you to imagine little voters. Maybe we have 5 molecules we're measuring for this person. And it may be that this one is above a critical value, this one is above a critical value, this one is not above a critical value, this one's above, this one's above. In a case like that, we have 4 voters saying aye and 1 voter saying no, and so we make the declaration, yes, this biomarker panel in this case is telling us that this person is really sick, right? Voting is a very simplistic way to combine together information of this sort. As we get to the machine learning part of this series, you're going to learn some much more complex methods that have been used for deciding which molecules get a vote and how that information can be combined quantitatively. Okay, this is my second favorite slide in this session. It's great to talk about biomarkers as an accomplished thing, that we already know all the molecules that we would want to measure in order to make any clinical decision out there. The reality is that there's a world of biomarker discovery still in progress out there. I want you to imagine that we have one funnel going this way, as shown on the slide, and we have another funnel going like this, starting very small and opening up very largely at the bottom. And we're traveling through both of these at the same time. In the one case, we're thinking about how many people we're measuring as we progress through biomarker discovery. In the first stages of biomarker discovery, we're looking at a very small number of people, maybe on the scale even of 100. At the bottom of that biomarker discovery pipeline, we're doing very broad testing in very heterogeneous populations spanning thousands of people. From a molecular point of view, the funnel points this direction. At the top of the funnel, at the start of the biomarker discovery process, we're testing thousands of different genes, or thousands of different transcripts, or thousands of different proteins. But at the bottom, we've filtered out all of the noise and gotten to just those proteins that are actually diagnostic for a particular condition. So if you are measuring thousands of proteins in samples, the number of people you're able to test, to do biomarker discovery in is a lot smaller. Like I said, like 100 or 200 people. As you get to just the top 5 proteins that are diagnostic for a particular condition, you've now transitioned out of the expensive technologies used at the top to something like a lateral flow test, and now we're testing just a very small number of molecules in a huge number of people. So you see that inverse relationship? Lots of people, tiny numbers of people, a very large number of compounds, down to huge numbers of people, a very small number of compounds. That's the inverse relationship. This is how we move from discovery of biomarkers to a confirmation, being able to do a second experiment that allows us to confirm that these proteins that we claimed were differential really are. Validation, to be able to evaluate the specificity and sensitivity, maybe even looking at different time points. These are people 6 months before diagnosis, these are people 4 months from diagnosis, these are people 2 months from diagnosis, these are people on the day of. Being able to use that kind of information for validation, and eventually working in clinical evaluation, being able to test how these tests work in a large population. Biomarker discovery is a wildly complex process. The technologies we use have to change. Maybe we start with mass spec, but end up with a lateral flow assay. There are many ways that we have to refine our hypotheses along the way. Maybe in this first test we found 100 proteins that looked differential, but we were only able to confirm that 30 of them really did change when we looked at them much more closely. And then we tried adapting to a different kind of technology for measuring those proteins using targeted proteomics rather than broad scale proteomics. And we found that some of those proteins were good for assay constructions and others were terrible. Maybe we had to move from biopsies in the first sample to biofluids in the next stage. That caused a lot of problems, and the proteins we thought would be very useful in fact are not. The biomarker pipeline is a very complex set of experiments, and it takes many, many steps to get from one to the other. Very, very frequently, we find that promising biomarkers at an early stage are of very little use when we start looking at them in a new cohort of people. As a result, although there's been a pile of money thrown into clinical biomarker research, there's been a lot of disappointment, particularly on the part of funding agencies that have thrown a lot of money at this technology. So, discovery to implementation failure. If I do a single experiment where I have 100 people who are sick and 100 people who are not, and I find a set of 5 proteins that magically let me discern all of the healthies from all of the 6, I might write a paper about that and be very self-congratulatory and say, I'm going to make piles of money because I've solved the problem of detecting everyone with tuberculosis in a population. Wouldn't that be great? You see a lot of papers, especially in early biomarker research, that make claims of near perfect discrimination that simply didn't hold up in retrospect. So it is difficult to do all of this transition, to prevent yourself from fooling yourself with your own data. Very, very challenging. It's wildly expensive. When we did a biomarker study using mass spectrometry for something like 100 patients at Vanderbilt, we spent 8 months of time on a single $1 million instrument. 8 months of its time was given over to just that examination of just these samples. When you have $1 million instrument with like a 5-year lifespan, 8 months of time is an amazing contribution of that instrument's time. So it's wildly expensive to do these things. They're certainly time consuming, not just for the instrumentation, but in trying to make good sense of the data that come back from them. There are lots of confounding factors that come into play. It may be that what we call the orange juice effect, that somebody drank orange juice on day 1 of the study, but on day 2 they didn't drink orange juice, and now the proteins that you thought were differential on day 1 don't hold up. That is an example of intra-individual variation. It may be that you have inter-individual variation. We know that people across South Africa have a wide variety of genotypes. This is a country of amazing diversity. And it may be that a test that works perfectly well in this particular subpopulation is completely irrelevant to people who have a different genetic background. Tissue localization is a big deal. It may be that a protein is hugely differential in ascites. Have people heard of ascites? A-S-C-I-T-E-S. It's a type of fluid that can develop in association with a tumor in cancer. It may be that a protein is hugely differential in ascites fluid, but completely non-differential in plasma or serum. Something like that can lead to great problems. It may be that the biomarker is around for a period of a few days, and if you don't happen to sample someone in that period, it's of no value. There's just a world of different problems that can cause this challenge. And so you find a number of papers coming out that say stuff like, the failure of protein cancer biomarkers to reach the clinic. Why is that happening? What can be done to address the problem? This is written by a friend of mine, actually. Pitfalls and limitations in translation from biomarker discovery to clinical utility in predictive and personalized medicine. We know we want to do this, but making it work is actually really, really difficult. Okay. So, we've covered a lot of topics, but I hope that this helps set the framework against which the other lectures will play. So a biomarker is a measurable characteristic that is indicative of biological state or condition. An ideal biomarker should be specific to that condition, should be sensitive, should be easily measurable, and able to differentiate different states. Bad prospect, good prospect for a particular condition. Biomarkers can be classified based on their application. Knowing something about each of these different types of biomarkers, I think, is really valuable to you. Biomarkers may be anatomical. You may measure them through imaging technologies or through a stethoscope. Or they may be molecular in nature. You may need something like an ELISA test for it. Panels that measure multiple biomarkers to make a decision offer improved sensitivity and specificity over single biomarkers, but they are considerably harder to figure out how to decide how big a biomarker panel you're including, for example. You may start with many candidates and end with, begins with many molecular candidates, and by the end you have just a few biomarker molecules you're actually going to include. At the same time as you've got this process of going from many potential molecules to very few, you have a very small number of people, or a relatively small number of people, going up to thousands as you get closer and closer to clinical applications. Okay, so we'll be back in here again on Tuesday at 10 o'clock when we will talk about immunoassays. So I will look forward to seeing you then. Thank you.